Hello again. Welcome to another episode of Fitness Facts. Dr. Jordan Taylor, the undergraduate exercise science program director and associate teaching professor here at the University of Kansas. So continuing on with this series on carbohydrates. In a previous episode that was Introduction to Carbohydrates. This is the second video in the series. This one today we're going to be discussing carbohydrate regulation during the fed state, so like after eating a meal, fasted state, and during exercise. So where are we going? This is an overview of what we'll be talking about. So we're going to get into the introduction to carbohydrate regulation in the body first. Then we'll discuss carbohydrate in the fed versus fasted state. Talk about hormonal maintenance of blood glucose, carbohydrates and exercise, how exercise intensity and duration affect fuel selection in the body, how long-term endurance training affects carbohydrate and fat oxidation during exercise, hormones and glucose metabolism during exercise, glycolysis, which is a pathway that our cells use to break down carbohydrates and produce energy, and then at the end, a little summary of exercise bioenergetic pathways. All right, so introduction to carbohydrate regulation in the body. So carbohydrate, primarily stored glycogen, which remember this can be, I discussed in a previous episode, stored in the muscles and the liver, and then also glucose, which is circulating around in the blood. They serve as essential but limited fuel sources in the body. So let me expand on that. So for instance, if you take an individual that's around 145 pounds and let's say they're 12% body fat, 12 to 13%, really lean, that's low body fat. They have, at that weight and 12 to 13% body fat, about 72,000 calories stored as fat in their body. You could survive on that for 3 weeks easily, 3 or 4 on that amount. If you're not doing a lot of activity and you're kind of just sitting and you're not moving around too much, but you easily survive 3 weeks just on your fat stores, your reserves of fat. However, carbohydrates are very limited, right? We only store about 100 grams in the liver as glycogen. We store another 400 grams or so in muscle, and then we have some of glycogen in muscle, and then we have some blood glucose, right? Those can be depleted very quickly if you're not eating and replenishing them, right? And I've talked about in a previous episode how 2 hours of intense activity can deplete those stores. Overnight, when you're sleeping, your liver glycogen levels become depleted. And that's why you need to break your fast. You eat breakfast in the morning to get your blood glucose levels or your liver glycogen levels back up. And the reason your liver's breaking down glycogen and releasing that as glucose throughout the night is it's trying to maintain your blood glucose levels. But overall, you just have a very limited supply of carbohydrate compared to, say, fat, okay? At rest, the liver, pancreas, and other organs help keep blood glucose levels within a narrow range to match the carbohydrate needs of the various body tissues. And we're going to talk about this, what happens in these different states, such as at rest, the fed state, the fasted state, and exercise. Glycogen is a vital energy source during muscle contraction, but this carbohydrate source is used more sparingly at rest. Most of the energy in the form of ATP, adenosine triphosphate, that we're producing at rest, a greater proportion or percentage of that energy is being produced from fat that we have stored on our body. We still are using some carbohydrate, but it's just the percentages. More of it is coming from fat to produce energy. As we start to transition to exercise and we're more active, now there's a greater reliance on carbohydrate, stored glycogen, also blood glucose, to provide energy at a more rapid rate, right? To provide ATP faster by breaking down those carbohydrate sources when we're exercising, okay? Adults store approximately 400 grams of glycogen in skeletal muscles, like I mentioned, and also another 100 to 120 grams of glycogen in the liver. Again, this is why we have this limited source, but we have even if you're really lean, like I said, a ton of fat stored on our body, and we can oxidize and burn that fat First, you have to release it from your fat cells, but then the fatty acids that are released in the blood go to your muscles, and your muscles can oxidize it and burn it for fuel to make and make ATP out of it. We produce ATP from glycogen breakdown at an accelerated rate during high intensity exercise. I mentioned that. We have less glycogen breakdown at rest and during, you know, like I said, lower intensity activities. But as we transition to higher intensity exercise, there's more glycogen breakdown. All right, let's look at fed state versus fasted state and define these first. So in the fed state, after eating, the body stores as much carbohydrate in the form of glycogen as possible while also stimulating carbohydrate fuel use to help return the blood glucose level to normal. Okay, so again, you're going to eat a high carbohydrate meal, you digest it, absorb it. Absorption occurs from the small intestine into the blood, and insulin's going to push that sugar into your various cells, and that's going to help return the blood glucose level down to normal. And also, that may stimulate some glycolysis, some glucose breakdown in the cells to again keep that blood glucose level stable. And that's in the fed state. The fasted state, this occurs approximately 8 to 12 hours after eating your last meal. So you're on an empty stomach here. What is the body going to do? Well, it mobilizes glucose precursors. So you can have some amino acid release, say from the muscles. You could have some glycerol being released from fat cells, and also, some fatty acids being released from fat cells. The amino acids and glycerol can circulate to the liver. The liver can take those, convert them into glucose. That's called gluconeogenesis, and then dump that glucose back out into the bloodstream to keep your blood sugar level stable between meals when you haven't eaten for, like I said, 8 or 12 hours. You don't want that blood sugar level dropping, or you're going to feel like garbage, okay? While promoting fat oxidation or energy to preserve carbohydrate as fuel. So that also happens in the fasted state. When you're not eating, not only is your liver making glucose from these non-carbohydrate sources like amino acids and glycerol, but your fat cells are going to tend to release fat, stored fat. Those fatty acids can travel to different cells like muscle cells, and they can be oxidized or burned to produce energy. When I say energy, that's adenosine triphosphate, ATP. So if we're burning more fat there, then we can preserve carbohydrate as a fuel source and save that until we would transition, say, to higher intensity exercise, okay? Liver glycogen levels, they definitely decline overnight because you're not eating. And the reason those levels decline, as I mentioned earlier, is that your body's trying to maintain an acceptable level of blood glucose. Like for the brain, some of your other vital organs, you need to have an adequate, stable level of glucose at all times. And the liver is the storage site of glycogen, and it plays a major role in releasing that and keeping your blood sugar levels stable, like between meals and also um, throughout the night when you're not eating. All right, this figure here, this chart on the left-hand side shows the fed state, short-term fasting, and long-term fasting, okay? The next column is the time after a mixed meal. So a mixed meal is like you ate some carbs, you ate some fats, you ate some protein. It's got all the different macronutrients in it. So from 0 to 4 hours after eating that mixed meal, you're in what's considered the fed state. Now, the third column is the blood glucose homeostasis or balance. What are the major sources of blood glucose in this fed state from 0 to 4 hours after a meal? Well, what's keeping your blood sugar up is going to be the carbohydrates you ate in that meal. Okay? So that's keeping your blood sugar level stable. Now, energy homeostasis and ATP production. So, what are the pathways inside your body's cells that produce energy in the form of ATP, adenosine triphosphate? How is that being produced, that energy in this fed state? Well, primarily glycolysis. So, again, sugar's entering cells from the bloodstream, glucose, and it can be burned through glycolysis to make ATP. Also operating on those cells are the TCA cycle, which is the Krebs cycle, an electron transport chain. We talk about that in my exercise biochemistry class. I'm not going to go through all the details here, but that TCA cycle and the ETC electron transport chain, they're important components of the mitochondria in our cells. They help to further break down carbohydrates, fatty acids, even amino acids to produce ATP in that fed state. Now, short-term fasting. So let's look at 5 to 16 hours after a meal. That's stage 2. How is blood glucose being maintained? What are the major sources of blood glucose during that 5 to 16 hours after meal? Well, liver glycogenolysis. So this is glycogen splitting, glycogen breakdown in the liver. So in the liver, at this stage, large stored glycogen molecules are being broken down, and then glucose can be released into the blood to sustain that blood glucose level in those 5 to 16 hours after a meal. Now, what are the major energy producing pathways? What are they doing in your different cells of the body, right? So, fatty acid oxidation is going to be ramped up here. You tend to be relying more on burning, oxidizing fat in the Krebs cycle and electron transport chain, the TCA cycle and electron transport chain, which occurs in the mitochondria of our cells, right? So, you start to go into this fasting state. Your fat cells will release fatty acids into the blood. Those fatty acids circulate to different cells in the body. The cells uptake them. The fatty acids, they enter the mitochondria, and your mitochondria, they can be burned and oxidized through the Krebs cycle and ETC. Now we move into 16 to 24 hours of fasting. Okay, this is still considered short-term fasting. Again, how is blood glucose being maintained? Primarily through gluconeogenesis and a little bit secondarily through liver glycogenolysis. There's less liver glycogenolysis occurring because if we've been fasting for almost a day, 24 hours, our glycogen levels are going to start to run low in the liver. So there's just not as much to break down, and there's not as much glucose to release into the blood to maintain our glucose level. So what the liver really ramps up here between 16 and 24 hours after a meal, where you've been fasting for almost a day, is gluconeogenesis. So the liver will start taking non-carbohydrate sources, amino acids like glutamine, alanine, glycerol, pyruvate, lactate, all these non-carbohydrate sources, and convert them into glucose and then dump that glucose back out in the blood to maintain a stable blood glucose, blood sugar level. Again, you're going to be relying heavily, if you look at the other cells of the body, they're going to be relying heavily on oxidizing and burning fat for fuel because you're not eating carbs. You don't have your primary energy source available. So you're relying on that fat oxidation for fuel. Long-term fasting. So if we look beyond a day, stage 4, and even beyond 2 days, right? Some people intentionally fast for religious reasons. Some people just fast for health reasons. There's some benefits with fasting there. But what's happening with long-term fasting? Well, in the liver, again, this is how you maintain blood glucose, gluconeogenesis. Some of this can also occur renal in the kidney, but this is cranked up. So you're producing approximately 130 grams a day in this long-term fasting. So again, using non-carbohydrate sources, glycerol, amino acids, lactate, pyruvate, converting those in the liver to glucose, and then dumping that glucose into the bloodstream to keep your blood glucose levels up. And again, as we see the theme here, short-term and long-term fasting, we see a lot of fatty acid oxidation, fat burning in our cells. And think about it. I watched that show, Naked and Afraid. It's on, I don't know, Nat Geo or Discovery, one of the channels. And people are on these islands and they haven't eaten for several days. And they're basically long-term fasting. They can't find food. And so how do they maintain energy production to keep walking around and looking for food or trying to get off the island? Well, they're going to release stored fat, and then they're going to oxidize and burn that fat in their muscle cells and other cells of the body to supply the energy they need to hopefully keep either escape the island, build a raft, find some food. You've got to rely on burning that stored fat when you're not taking in any food, okay? Also, ketone oxidation ramps up with long-term fasting. So when you've been fasting for over a day, maybe 2-3 days, you are releasing a lot of stored fat as fatty acid into the blood. Some of that fatty acid goes to our cells and gets oxidized and burned to make ATP. Some of those fatty acids go to the liver, and the liver can convert them to ketones, and then those ketones can be dumped back out into the blood. And a lot of different cells and tissues can use ketones for fuel, like the brain, the heart, our muscles. And that can keep us going until we ultimately find some food and eat again. All right, so let's briefly kind of review the hormonal maintenance of blood glucose. So, hormones, there are really several of them here that have a major impact on either raising or lowering um, blood glucose. So, in the well fed state, right after a meal, assuming that meal had a lot of carbohydrates in it, um, some amino acids also raise insulin, but carbs, amino acids that are entering the blood, that's going to cause the pancreas to release insulin. Okay. Insulin, what's the response to that in the well fed state? Insulin knocks on the doors of cells, muscle cells, fat cells in the liver, and it binds to receptors on those cells, and then those cells uptake glucose out of the blood. Okay, so it reduces glycemia, meaning insulin's going to reduce the presence of glucose in the blood because the glucose gets shuttled into your cells. Also, insulin stimulates lipogenesis. So this is fat storage, okay? And it increases proteosynthesis, which is the synthesis of proteins. Okay, that's another function of insulin. You can think of it as like an anabolic hormone. It's telling cells to build protein. Um, Fasting state. So this is again between meals. You're not eating. Even overnight, like when you wake up in the morning, you're in a fasting state. So what hormones are going to be high in the blood? Well, glucagon is going to be high. That's released from the pancreas when you're in a fasting state. Adrenaline, there's epinephrine, and then there's also norepinephrine. Those are released from the adrenal glands that sit on top of the kidney. And cortisol, which is also released from the outer part, the adrenal cortex of the adrenal gland. So those hormones, glucagon, adrenaline, which is epinephrine, and you also have cortisol, they're elevated in a fasting state. And when they're elevated in the blood, what's the response of the body? Well, they all work to increase glycemia or increase the amount of blood sugar or the amount of glucose that's in the blood. Okay. So glucagon, cortisol, epinephrine, these can all tell the liver to break down glycogen and release that after the glycogen is broken down, release glucose in the blood to raise your blood sugar level back up. Okay. Um, they also signal for increasing lipolysis, that's fat breakdown and release. Ketogenesis, the formation of ketones, which I talked about, are an important fuel source in a fasted state. And in a fasted state, these hormones play a key role in proteolysis. So this is where, if you haven't eaten for 2 or 3 days, you're stranded on an island. You're going to not only burn fat for fuel, but you're going to start losing some muscle mass because you aren't taking in calories. And these hormones that are high in the blood signal protein breakdown. So muscles are primarily protein. They will release amino acids into the blood. And then the liver will take those amino acids, and this is all directed by these different hormones, and convert some of those amino acids into glucose and dump that glucose back out into the blood to keep your blood sugar level stable until you actually eat again. Okay. Now, if we look at insulin and glucagon, so two of these hormones that are really important in the fed state and fasted state for regulating and controlling our blood glucose level. So follow these arrows with me. So if you have high blood sugar, when would this occur? After you eat like a high carbohydrate meal. Okay. What happens? So high blood sugar, fed state, that promotes insulin release from the pancreas. Okay. The pancreas, you follow the next red arrow, you see insulin released, that stimulates glucose uptake from the blood into our tissue cells. So insulin stimulates the uptake of glucose into the muscle cells, the liver cells, fat cells, which in turn is going to lower blood sugar. Okay. Insulin also stimulates the liver to form glycogen. So glucose can be many glucose molecules linked together and stored as glycogen in the liver. That's under the direction of insulin. Okay. Now what happens the opposite? If we follow the green arrows, okay, if you're in a fasted state, your blood sugar is going to start to maybe drop and be lower, okay? So that promotes glucagon release from the pancreas. Glucagon, when it's released from the pancreas, is going to stimulate the liver to break down glycogen and convert that glycogen to glucose. And then the glucose can be released into the bloodstream, which will raise blood sugar back up to a more normal level. So this is kind of the yin and yang of blood glucose control, right? Insulin's function is to lower blood sugar. Glucagon's function is to raise blood sugar. And then also other hormones released from different glands like cortisol, epinephrine, norepinephrine. Um, those all have a role too in increasing blood sugar, as I mentioned on the previous slide. All right, so this slide, again, more on the hormonal control of metabolism. On the left-hand side, you see a couple graphs there. The kind of purple line is glucose level after eating a high carbohydrate meal. Then the middle graph, so that's concentration of glucose in the blood. Then the concentration of insulin in the blood. And then the concentration of glucagon. So you see that after a high carbohydrate meal, glucose levels go up in the blood really 30 to 60 minutes, they get pretty high. Insulin also is being released from the pancreas and its level in the blood rises. But after that high carbohydrate meal, glucagon goes down, right? These are hormones have opposing actions, so you wouldn't want glucagon and insulin being high at the same time. That would really create an issue there with signaling, okay? Because insulin's trying to lower blood sugar, glucagon's trying to raise it. So again, insulin increases after that meal. That's going to help shuttle glucose into your liver cells, fat cells, and muscle cells, and glucagon will stay low. If we look at the four major molecules that control carbohydrate homeostasis and overall metabolic homeostasis, again, we have them listed there: insulin, glucagon, epinephrine, and cortisol. Okay, so the function of insulin, of course, promote fuel storage after meal. It helps promote growth through like protein synthesis. And I have arrows there in red that show their major functions of each of these hormones as it relates to carbohydrate, okay? I mean, these hormones have different functions related to fat, amino acids, protein synthesis, but I'm just kind of highlighting, since this is talking about carbohydrates, the red arrows highlight each of these hormones' actions with regard to carbohydrate. So insulin stimulates glucose storage as glycogen in the muscle and liver. Glucagon, as I mentioned, it can activate gluconeogenesis in the liver, which is forming glucose from non-carbohydrate sources like amino acids, glycerol, lactate, pyruvate. And it also activates glycogen breakdown in the liver during fasting. And this all helps to raise blood sugar levels, because when you break down glycogen in the liver and you create new glucose, all of that glucose can be released into the blood during times between meals and states of fasting. Epinephrine stimulates glucose production from glycogen. So again, the liver can release that stored glucose out into the blood. Muscle, however, cannot. Once glucose is enters muscle, it gets a phosphate group tacked onto it and becomes glucose 6-phosphate, and it gets trapped in muscle. And so there's no way of releasing that. But it can get glucose can get created from glycogen in muscle, and then that glucose can stay in that local muscle cell and go through the glycolysis pathway, and you can make ATP. Okay. So the liver is really the main organ that releases glucose from stored glycogen into the blood. Basically, any glucose in muscle is trapped there. It either gets burned or it's stored as glycogen in muscle. And then cortisol, it stimulates gluconeogenesis. So cortisol will tell the liver, knock on the liver's door, and say, hey, blood sugar levels are low. You need to start making some glucose out of amino acids or glycerol or pyruvate or lactate, and then dump that glucose that you make from those sources back into the blood. For instance, cortisol, if you wake up in the morning, it tends to be pretty high in the blood. And it's signaling the liver to dump out and release glucose to get your blood sugar level up so you can get out of bed, get your clothes on. Maybe you got some errands to run, but it provides you some fuel before you even eat breakfast or maybe you don't eat breakfast and you wait till lunch. You got to have your blood sugar level stable during that time. Cortisol helps direct that. All right, so during exercise, the body increases the use of fat and carbohydrate as fuel needs rise, right? When you're exercising, your body begins to break down glycogen stores in muscle and uptake and also break down glucose from the blood, right? Now, the process known as glycolysis is an enzymatic pathway. There's 10 steps in this pathway our cells use to break down glycogen and glucose to create ATP or energy, right? So the breakdown of glycogen and glucose in that pathway to produce energy is glycolysis. To maintain blood glucose levels during exercise, the liver increases the rate of hepatic refers to the liver, the rate of glycogenolysis, which is the rate of liver glycogen breakdown, and it also ramps up gluconeogenesis. Okay? The amount and ratios of carbohydrate and fat used during exercise depends on several factors. Fat is the primary fuel source when at rest and during low to moderate intensity activity. Okay, so fat released from our fat cells, fatty acids being released from those fat cells helps to fuel the cells of our body at rest and also when we're doing low to moderate intensity prolonged exercise. Carbohydrate, however, both the stored glycogen in muscle and also glucose that could be coming in from the blood into muscle, there's a greater percentage of its use when we increase the intensity of activity that we're doing or we increase exercise intensity. So if we move from low to moderate exercise and we do more high intensity exercise, like say we move from a walk to all of a sudden doing sprints, yeah, we're going to have a greater reliance there during the sprints on carbohydrate breakdown, glycogen and glucose. If exercise continues for prolonged durations, glycogen stores in muscle and the liver decline, resulting in a need to shift back to fat as a significant source of fuel. However, if you have to rely heavily on fat as a fuel source during exercise, your exercise intensity will be reduced. And that happens because it takes much longer to release fat from fat cells, circulate it in the blood to muscle. It has to be uptaken into the mitochondria. It has to go through a process called beta oxidation and then through the Krebs cycle, then through the electron transport chain. It's a much more lengthy, long process to derive energy in the form of ATP from fat than it is glycogen that may be readily available right there in the muscle. So because it takes such a long time to derive energy from fat, you can't maintain the same race pace or the same intensity of exercise because you just can't supply ATP energy as fast when you're trying to break down fat versus glycogen or glucose. You can break that down much more quickly. There's less steps involved. You can supply ATP, that energy your cells need, at a much more rapid rate. And that way your exercise intensity can stay high. This shows fuel selection, I think, very nicely during exercise. So as I mentioned, carbohydrate and fat can both be main fuel sources during exercise. Now, the proportion that you use of carbohydrate versus fat is going to be dependent on exercise intensity and the duration of exercise. So as exercise intensity goes up, we use relatively more carbohydrate. Okay, so you see at rest there, and exercise intensity on the x-axis is listed as a percentage of your maximal workload. So at rest, then you have 40% of your max workload, which would be lower intensity, 55% is more moderate. Then once you get to 75 and above, that's more heavier intensity exercise. So at rest, most of the fuel for the body is coming from fat, free fatty acids coming into cells from the blood, and also some glucose, so some carbohydrate. At low intensity exercise, you see fat in black. You see fatty acids, another source of fat, in gray. And in white, you see some plasma glucose. And you start using a little bit more muscle glycogen. But again, predominantly, it's mainly fat sources there in the black and gray. So fat is being used as a fuel for low intensity exercise. Then as you move to more moderate intensity, you still see a lot of gray and you see black there, meaning that you're using a lot of either free fatty acids floating around in the blood, fat being released from fat cells. You're using fat, oxidizing that as a fuel source. You have increased the exercise intensity a little bit, going from 40 to 55. So you are using some more muscle glycogen, carbohydrate there. Now, once you get to higher intensity, 75 and above, you see that there's a lot more green. We're relying a lot more on muscle glycogen there to fuel that higher intensity activity, less so on the free fatty acids and other fat sources. The exercise crossover concept. Okay, this is an important concept to understand. This just shows that during low to moderate intensity exercise, fat and carbohydrate, yeah, they both play a role, as we mentioned and you saw in the previous graph. But when aerobic exercise power output increases to above 65% of VO2 max, carbohydrate becomes increasingly important as a fuel source to generate ATP for muscle contraction during higher intensity exercise. So you might wonder, well, what is VO2 max? So on the x-axis, the horizontal axis on the bottom, you see the percentage of maximal oxygen uptake, okay? At rest, you're only taking in about 3.5 milliliters of oxygen per kilogram of body weight per minute. As I would go from, say, resting, sitting here, and if I got up and started walking around the room, or maybe I went out in the hall and started running, the percentage of oxygen that my body takes up is going to increase, right? So I'm breathing in more oxygen at a more accelerated rate. My respiration rate's up. More oxygen is leaving the lungs and entering the bloodstream, and oxygen is being circulated to my working muscles at an accelerated rate during exercise. So I'm consuming more oxygen and my cells are taking it in at an accelerated rate during exercise. And they have a max capacity for doing this. That's called your VO2 max, basically the maximal level of oxygen consumption by your cells that you can achieve during exercise. And that oxygen is important in your cells because it helps to oxidize and burn fatty acids. It helps to also oxidize and burn glucose and glycogen. So you can measure exercise intensity by measuring a person's oxygen uptake. And you know that they're exercising more intensely as oxygen uptake percentage goes up, okay? And also what's occurring as oxygen uptake goes up is your heart rate is higher. So you can just imagine if you go along the x-axis and you go from rest to 20 to 40 to 60 to 80 to 100% oxygen uptake, your heart rate is going up along that whole way too. So at higher heart rates and higher oxygen consumption rates, you're going to see that you use a greater percentage of energy is derived from carbohydrate. At lower percentages of oxygen uptake and lower heart rates, a greater percentage of energy is being derived from the burning of fat, okay? So the blue line is fat. You see at rest, a lot of our energy is derived from fat, and at lower intensities, less from carbohydrate. But as we move closer to that crossover point, which occurs at about 65% of VO2 max, that's a moderate exercise intensity. At that point and above, that's where you really start to rely more heavily on energy derived from carbohydrate. So if you look at the right side of the graph, y-axis, vertical axis, the percent of energy derived from carbohydrate, that's an ascending scale. At the bottom, it's 0, then it goes up to 10, 20, 30, 40, all the way up to 110. As you move past the crossover point, you see how that red line's increasing. We're really using much more carbohydrate, but the amount of fat we're using is dropping off, okay? And the fat, energy derived from fat, is on the left-hand side vertical axis. So that's again, from the bottom, 0, 10, 20, 30, going all the way up. So you're using less fat with higher intensities, more fat with lower intensities. So that crossover point is just where this shift really occurs. Now, training, interestingly, if you do a lot of aerobic endurance training over weeks and months, that training will move that crossover point to a higher percent of your VO2 max. Meaning, if that crossover point shifts to the right, you will be oxidizing and using fat at a greater percentage of the time, and you're going to spare carbohydrate. You're really not going to start using carbohydrates until very high intensity exercise. So it's like training improves your ability to spare and store carbohydrate, and get more efficient at burning fat because it shifts that crossover point to the right. Okay. Now, the question is, and I get this a lot, I used to get this from clients when I was a trainer. Students ask this, well, should I only exercise at lower intensities if I want to burn more calories from fat? Because on that previous graph, you saw that a greater percentage of your energy was derived from fat burning at lower exercise intensities. So maybe if I want to burn a lot of fat, I should just, go on, slow jogs or a light walk, keep my exercise intensity low because I'm going to burn a greater percentage of calories from fat. That is true. However, let's break this down further. The answer really is no here. It's okay to do low intensity exercise and you need to. That should be part of your training. But if your goal is burning fat, should you always do low intensity exercise? Maybe not. So let's look at this. So in this table, we're comparing caloric expenditure, the percentage and amount of carbohydrate and fat used during 30 minutes of jogging at high versus lower intensity of jogging. And this is for someone that weighs 150 pounds. So looking at calories expended, and then what percentage of carbohydrate and fat is used during 30 minutes if this person that weighs 150 pounds, we're going to jog one day at a low intensity and one day at a high intensity. So the duration's the same, right? They're going to jog for 30 minutes, but one day it's low intensity, one day it's high intensity. And you see at the lower intensity, they're only covering, they're covering 10 minutes per mile, or 1 mile in 10 minutes, right? So that's lower intensity. You're running at a slower, jogging at a slower pace. Higher intensity for 30 minutes, that session, they're covering each mile in 7 minutes. So their pace is faster, right? Now, let's look at the second row, calories per minute burned. Well, this makes sense intuitively. When training for 30 minutes at the lower intensity, they're burning approximately 12 calories per minute. At the higher intensity, approximately 17 calories per minute, right? You exercise at a higher intensity, burn more calories. Lower intensity total calories burned in 30 minutes, about 360 for this person. And at a higher intensity, 510. Now, again, the total amount of calories you burn, there's a lot of factors that go into this. Your weight, if you're how trained your training status. So there's a lot of factors that contribute to this. But for this person weighing 150 pounds, you see the numbers there. They did burn more total calories during the 30 minutes of high intensity activity. Now we're getting into the nitty gritty here with the percentages. At the lower intensity, of the 360 calories burned, 48% came from carbohydrate, 52% came from fat. Okay? So, yes, we did burn a greater percentage of calories from fat at the lower intensity. Okay? Now I'll go over to the far right column. High intensity exercise, they burned 510 total calories. Of course, at higher exercise intensities, a greater percentage of carbohydrate was burned, so 60%, while only 40% of the energy was derived from the burning of fat at that higher exercise intensity. But let's look at amount of carbohydrate as far as calories burned and amount of fat, okay? This is where I think it becomes clear. At lower exercise intensity, there were 173 calories of carbohydrate burned and 187 calories of fat. We go over to the right-hand column. At higher exercise intensities, yeah, a lot of carbohydrate burned, 306 calories, but there were 204 calories of fat burned, okay? So although the percentage of fat burned for the high intensity exercise day was less than the low intensity day, there were still more calories burned during that from fat during that high intensity exercise bout. It's just the percentage was lower, but you still burned more fat calories during the 30 minutes of higher intensity exercise, 204 versus 187. Okay. So I think that makes it a little more clear that, yeah, you can look at percentages, but we also have to look at amount of carbs versus fat burned. Okay. So this graph, again, prolonged exercise necessitates the increased use of fat for fuel. There's less reliance on carbohydrate with increasing exercise duration. So again, Exercise intensity. Low exercise intensity, greater percentage of fat burned, less carbohydrate burned. High exercise intensity, greater percentage of carbs burned, less fat burned, but you still may burn an overall greater amount of fat, assuming you could exercise at that higher intensity for the same amount of time, same duration as the lower intensity exercise bout. Now here, this is showing exercise duration. So you go on the y-axis from 0 out to 20 minutes, 60, 100, 120 minutes, right? 2 hours of exercise. And then on the y-axis, the vertical axis, you see what's the percent of fat or carbohydrate that's being metabolized, broken down to provide energy. Well, over time, if we look at the pink line, you see that the longer we exercise, the more our body has to rely on fat as a fuel source to produce energy. Part of this is because glycogen levels are being depleted in muscle, so our carbohydrate in muscle is going down. The longer we exercise, glycogen in the liver is being depleted. So again, our carbohydrate stores are depleting, especially if we're not replenishing them by drinking like a sports drink or some carbohydrate gels if we're going to exercise for a long period of time, okay? So carbohydrate reliance is less with increasing duration as seen on the descending blue line, all right? So over time, less reliance on those carbohydrates with prolonged duration. Now let's talk about hitting the wall or bonking. So hitting the wall is a condition of sudden fatigue and loss of energy, which is caused by the depletion of glycogen stores, so carbohydrate in the liver and muscles, during prolonged endurance exercise. Okay, now let's switch over to the right. So here you see energy sources that are used in a working muscle. And this is the x-axis, the horizontal axis, time of exercise over minutes. So from 0 to 100 minutes to 200 minutes to 300 minutes of exercise. So that's a long time. Okay. You see the wall, that big red line, that's indicating, that's at about what, 240 minutes of exercise. So that's, 60 minutes in an hour times 4 hours. That's 4 hours of exercise. Okay. Right there, this individual has hit the wall, meaning their muscle glycogen levels have dropped off and are depleted. Their blood glucose levels are declining because the liver glycogen levels have declined. So the liver's having trouble uh, releasing glucose into the blood and keeping blood glucose levels up. So you're seeing that, like carbohydrate sources of energy are going down because they're being depleted during this prolonged exercise bout. While the dotted line, fatty acids, has increased, right? We're releasing more fat from the fat cells, having to rely an increased reliance on that fat for fuel. Okay, typically in a marathon, if runners don't adequately supplement with carbohydrate through that race, and I'm going to talk about this in another episode, how you should supplement with carbohydrate during exercise so you don't hit the wall. Typically, if runners are going to hit the wall because they can't maintain their muscle glycogen levels, their blood glucose, and their liver glycogen, they typically hit it around mile 20. All right. And part of this is, if we do the math, the reason they hit it at mile 20 is, let's assume, well, first let's talk about, remember that there's 100 grams of glycogen stored in the liver, on average, 400 grams of glycogen stored in muscle, for a total of 500 grams of glycogen or carbohydrate stored in the body. Each gram of glycogen is worth 4 calories, okay? So if there's 500 total grams of glycogen stored in the body, and we multiply that by 4 calories per gram, we have 2,000 calories of energy available to fuel us, okay, in the form of carbohydrate, 2,000 carbohydrate calories. Depending on the person's weight and the intensity they're running at, let's just say they're burning around 100 calories per mile, okay? And if those calories are coming from carbohydrate, initially, think about this. You're burning 100 calories per mile. 100 calories per mile times 20 miles, that's 2,000 calories. How much carbohydrate as glycogen did I say stored? You have stored 500 grams, and there's 2,000 calories of carbohydrate available for the body. So if you're burning 100 calories of carbohydrate per mile and you hit mile 20, you've went through all 2,000 calories, okay? And that's where you could hit the wall. This is why it's so important in prolonged endurance exercise to supplement with carbohydrate so you don't hit the wall, because you're going to feel at this point, when you have severely depleted your carbohydrate stores, very fatigued, weak, your race pace is going to drastically have to drop. Your intensity of running is going to significantly drop, maybe to a walk. You may even have to stop, okay? Because you just can't maintain the same intensity because of the depleted carbohydrate stores, glycogen, blood glucose, liver glycogen, they're all down. Okay. So really the only thing you have to fuel is fatty acids, fat oxidation. Okay. Beyond that point. So this condition, hitting the wall, can usually be avoided by ensuring that glycogen levels are high before the exercise begins, and then also maintaining glucose levels during the exercise by eating or drinking carbohydrate rich substances, or by reducing exercise intensity. So, that's where it's important to pace right if you're running a marathon, right? If you start out too high intensity, running too fast early on, you could drastically reduce your carbohydrate stores, glycogen levels, and the onset of fatigue could occur sooner, okay? All right, so we'll go to this next slide here. This is looking at carbohydrate and fat oxidation during exercise in untrained individuals versus endurance trained individuals. So we're comparing somebody that's untrained, somebody that doesn't exercise, doesn't do a lot of running, versus somebody that's, let's say, has been doing a year of endurance training, okay, or even 6 months. All right, what is this graph showing? The untrained individuals are the dark black circles and that dashed line of best fit. Okay, that's the data for untrained. The endurance trained individuals, done weeks and months of endurance training, are the open circles and that solid line. Okay, exercise intensity is shown on the x-axis, the horizontal axis at the bottom, as a percentage of VO2 max. Remember, at lower percentages of VO2 max, like 40%, that's lower intensity exercise. When you get to 60 to 75%, that's moderate intensity exercise. Once you get into the 80, 90% of VO2 max, that's higher intensity exercise, okay? On the left vertical axis, you see carbohydrate oxidation, so the percentage of total oxidation of carbs on the left vertical axis. On the right vertical axis, y-axis, we're looking at the percent of total oxidation of fat, okay? Now you notice the carbohydrate oxidation on the left, it's going up it's ascending from 30, 40, 50, 60, 70, 80, 90%. On the right vertical axis, we have descending. So going from 10 down to 20, 30, 40, 50, 60, 70, okay? But basically what's shown here is that trained individuals that are endurance trained, they've been doing this for months, 6 months to a year. Their line has been shifted down, okay, and to the right, which means that at any given exercise intensity, low, moderate, and even high, they're using more fat as a fuel source than if they were untrained. Okay? So as you become more trained, your body learns how to utilize fat and burn it more efficiently as you go through the months of aerobic exercise training, running, jogging, swimming. You get more efficient at burning fat at all these different exercise intensities. There's a greater rate of fat oxidation occurring. So you burn fat more efficiently, and you learn how to spare muscle glycogen and liver glycogen. So you rely a little less on it and you save more of it. Okay. However, untrained individuals, they tend to rely more heavily on the breakdown of carbohydrates, muscle glycogen, liver glycogen, until they become more trained, right? And then their body learns to use fat more efficiently as fuel. Okay. All right. So hormones that increase glucose metabolism during exercise. So I mentioned glucagon. A lot of these right here that you see listed that increase glucose metabolism during exercise, they also are elevated in the blood when you're fasting, okay? So glucagon is released from the pancreas. You see a picture of the pancreas there on the right, released into the blood from the pancreas. Epinephrine is released into the blood from the adrenal gland medulla. That's the inner part of the adrenal gland. So the adrenal gland looks like a little hat that sits on top of the kidneys. You have two of them, one on each kidney. Norepinephrine is also released from the adrenal gland. And then you have cortisol that's released from the adrenal gland cortex. That's the outer layer of the adrenal gland. All of these are released and they're in elevated levels in the blood when you're exercising, all right? And what they do is help to stimulate glycogen breakdown in muscle. They help to stimulate glucose formation in the liver and the release of glucose from glycogen in the liver back out into the blood to help maintain blood glucose levels. Now, if carbohydrate reserves are running low, so you've been exercising for a long period of time, you're depleting muscle glycogen, depleting liver glycogen. These hormones, cortisol, norepinephrine, and also growth hormone, which isn't shown here, growth hormones released from the pituitary gland, those hormones can also all work together to stimulate the fat cells to release fat as fatty acid. That fatty acid can circulate in the blood to the muscles, and then the muscles can oxidize it and burn it to produce energy. Now let's look here at blood glucose levels after sprinting. So we're going to look at different forms of exercise and how it affects carbohydrate metabolism. Okay, so the concentration of glucose is on the y-axis, vertical axis, and then the 60-second sprint from minus 60 to 0, time 0, is shown there on the graph. So that red arrow is indicating that 60-second sprint. Sprinting is a high-intensity exercise, right? Anybody that's sprinted knows that. So you do this high-intensity exercise, this 60-second sprint, and what happens is epinephrine and norepinephrine release occurs from the adrenal medulla. So these hormones, epinephrine and norepinephrine, rise in the blood. And what do? They cause blood glucose concentrations to temporarily increase during this short duration, high intensity exercise because they signal glucose to be released from the liver at a greater rate than what the muscles can uptake the glucose from the blood is occurring at. So you actually see after sprinting, due to the high levels of epinephrine and norepinephrine in the blood signaling the liver to release glucose into the blood, you have this little spike in blood glucose. And then after the sprint, typically those blood glucose levels are going to go back down. Muscle can uptake it. And so you see the levels drop pretty rapidly after in recovery from the sprint, the rest period after the sprint. All right, let's look at plasma levels of hormones during cycling at 65% of VO2 max. So again, this is like a moderate intensity. So moderate intensity cycling over long duration. So you look at this, time is on the horizontal axis from time 0 out to 180 minutes. So that's 3 hours of cycling. And then we're looking at the percentage change. So how these hormones increase in the blood on the vertical axis. Okay, so again, cycling at this moderate intensity. First, let's look at the blue line that stays pretty much flat over the 3-hour time period. That's blood glucose. So, yeah, it's like you might ask, well, yeah, how does your body maintain blood glucose over 3 hours of cycling since glucose is a source of fuel along with the muscle glycogen and the liver glycogen? How are we maintaining that? Hormones are doing it. So, you see cortisol levels rise from about 15 minutes to 30 minutes of cycling. Cortisol, again, along with these other hormones, are telling the liver to create more glucose from non-carbohydrate sources and then to release glucose from stored glycogen in the blood to keep that glucose level flat and stable over the 3 hours. Glucagon, you see the green line, it rises over the 3-hour time frame and kind of flattens off at 60 minutes. It's the green line. Epinephrine and norepinephrine. So epinephrine is the black line, norepinephrine is the red line. You see those hormones steadily increase in the blood all the way out to 3 hours, 180 minutes. But again, all four of these, epinephrine, norepinephrine, glucagon, cortisol, they're really just working together synergistically to maintain blood glucose levels. And they're really telling the liver what to do. Like, hey, release glucose into the blood and make more glucose so that your blood sugar level stays stable. Because what's happening if you're cycling for 3 hours? Your muscle glycogen levels are going down. You're burning glycogen, that stored carbohydrate in muscle. So you need to uptake glucose from the blood As the muscle glycogen levels go down, that glucose coming into muscle from the blood during this prolonged exercise can be broken down to make more energy, more ATP. Okay. And again, these hormones just play a role in keeping that blood glucose level up by directing the liver, telling it what to do. All right, this slide shows plasma levels of glucose and insulin during prolonged cycling at, again, a moderate to slightly higher intensity. So 65 to 70% of VO2 max. That's still a moderate intensity. So there's a gradual decline in insulin throughout exercise at this moderate intensity. However, what's really cool is your muscle cells actually become more sensitive to insulin, even though its level declines in the blood. So therefore, glucose can still enter muscle cells even though your plasma insulin level is lower. Remember how I've said, like after a meal, insulin level goes up, insulin tells your cells to uptake glucose. Well, in this case, during exercise, insulin goes down. So you might be thinking, well, then how's the glucose going to get into the cells? Well, contraction, muscle contraction allows it to come in, but also, even though the insulin level is lower, the receptors for insulin and the muscle cells themselves are just more sensitive to it. So less insulin is available and less insulin is needed to promote glucose uptake from the blood into the muscle. All right. I've talked about this in a previous lecture, but I want to kind of rehash it and go over it again here. Yes, insulin facilitates, if you look on the left-hand side, when insulin binds to its receptor, it activates an intracellular signaling cascade. So there's a bunch of proteins and enzymes that are all different colors there, kind of the different color circles and ovals, orange and green and yellow. Those get switched on. There's a lot of phosphorylation of those proteins and enzymes occurring. And then you follow the arrows. There's this signaling cascade, kind of this domino effect as those get switched on that causes these GLUT4 transporters, these are glucose transporters, to move from inside the muscle cell to the muscle cell membrane. And there you see 3 of those red glucose transporters embedded in the muscle cell membrane in response to insulin binding to its receptor and activating this signaling cascade. Once those GLUT4 transporters are in the cell membrane, they provide a doorway for glucose to flow into the muscle cell from the bloodstream. And now you've got that glucose available to be broken down, and you can produce ATP energy from it to keep fueling muscle contraction. On the other side, the right side of that dark black vertical bar, you see muscle contraction. So muscle contraction also stimulates the uptake of glucose through those GLUT4 transporters, right? So when muscles are contracting, the sarcoplasmic reticulum, which is this basically storage house of calcium inside a muscle cell, it starts to release calcium. Calcium can signal different things. It signals calmodulin and it signals AMPK. They're in yellow. And that calmodulin and AMPK signal the GLUT4 transporters to move to the cell membrane and create doorways for glucose to flow into the cell. Okay. So again, both muscle contraction and insulin, through different signaling mechanisms, cause the GLUT4 transporters to move to the cell membrane, and then glucose can move into the cell. So you've got that fuel available to keep exercising, all right? This slide is cool because, it's talking about exercise sensitizing muscle cells to insulin, okay? This makes it easier to uptake glucose from the blood into muscle, which helps lower blood glucose levels. And this is why exercise can be so useful for any of you that have type 2 diabetes and you're looking to control your blood glucose levels. So just one bout of exercise can have this effect. And if you exercise over weeks and months, your cells even get more sensitive to insulin and they get even better at uptaking glucose out of the blood to help lower and stabilize your blood sugars, right? Because you don't want high blood sugar levels. It's damaging to your cardiovascular system. It can be damaging to the retina in your eye. It can be damaging to your kidneys. It creates damage everywhere, okay? So exercise can help stabilize and reduce it. All right, so I'm going to go to the bottom right-hand corner there. If we look at rested muscle, there's 2 different types of cells. There's an endothelial cell, which lines blood vessels, and then there's a muscle cell shown, muscle fiber also, okay? Insulin can bind to receptors on both these cell types, the endothelial cells that line blood vessels, and there's insulin receptors on the muscle fiber, Okay, so you've got some insulin binding there to the receptors in a rested muscle. In the endothelial cell, that insulin will cause phosphorylation of an enzyme called endothelial nitric oxide synthase. So phosphorylation, you see that little P inside the yellow circle. When that endothelial nitric oxide synthase is phosphorylated and insulin binding to the receptor directs this, you're going to get some nitric oxide released from the endothelial cell. Nitric oxide is a gaseous molecule, and what nitric oxide does is allows for some vasodilation to occur, which could improve glucose delivery to the muscle. So that in the muscle, insulin binding to the receptor there through TBC1D4. This is a protein inside the muscle cell. Insulin will activate that, and the TBC1D4 will instruct those glucose transporters to move to the cell membrane and uptake glucose. And then that glucose can enter the cell from the blood, and the glucose molecules can be linked together, and you can store that glucose as glycogen. Okay. But what happens in an exercise that is, or a muscle that's been has just underwent exercise? It's been previously exercised. These signaling pathways are enhanced. Okay. So again, muscle's just been exercised. It's more sensitive to insulin. What's going on in the endothelial cell? Well, insulin binds to the receptor. Also, exercise has stimulated an enzyme called AMPK. So exercise switches on AMPK in the endothelial cell and also in muscle. Okay. Insulin is binding to receptors because these cells are sensitized to it during exercise. And in the endothelial cell, you can see that the combination of exercise switching on AMPK, insulin binding to the receptor, causes even greater phosphorylation of that endothelial nitric oxide synthase and even greater nitric oxide release. You see more nitric oxide molecules there being released from the endothelial cell. This causes a greater amount of vasodilation in the blood vessels and the capillaries going to the skeletal muscles. Because the blood vessels are dilated more, they're more open, you have greater blood flow and glucose delivery to the skeletal muscles. How does that glucose get into the muscle fibers? Well, exercise, muscle contraction activates AMPK. The cells are sensitive to insulin. Insulin binds to the receptor. And the activation of AMPK and insulin binding to the receptor causes that phosphorylation. See the little yellow circle with the P on it, of the protein TBC1D4. When that protein is phosphorylated and switched on, you get even more GLUT4 translocation. You see there's 4 glucose transporters embedded in the muscle fiber membrane. So there's more doorways for glucose to enter the cell, the muscle cell, from the blood, because there's 4 of them in purple, 4 glucose transporters embedded in response to exercise and insulin. Because that muscle fiber is sensitized, it's easier for glucose to get in, and it's easier for you to store that glucose as glycogen in the muscle cell as a result of that muscle being exercised. Whereas you go back down to the rested muscle, there's only 2 glucose transporters embedded there in response to insulin, but the muscle hasn't been exercised, so we don't get the AMPK activity, we don't get the extra nitric oxide release, so there's just less glucose entering into that muscle. And as a result, there's less muscle glucose uptake, which is represented on the graph on the left. So skeletal muscle glucose uptake is on the y-axis, insulin on the bottom. You see in rested muscle, for any given level of insulin, there's less glucose uptake. That's the red line. But then the curve shifts to the left and up, the blue line. If the muscle has been exercised for any given level of insulin, there's going to be more glucose uptake. The muscle cell has become sensitized to insulin, and there's more glucose transporters available, so it's easier to get that glucose into the muscle fiber for any or the same given level of insulin if you're comparing the exercised muscle to a rested muscle. All right, also showed this in a previous lecture, but just for everyone to be clear on it, once glucose enters a cell through those glucose transporters, so you see glucose in yellow, entering from the blood through the red glucose transporter. An enzyme called hexokinase is going to tack a phosphate group onto that glucose molecule at the 6th carbon position, and it becomes glucose 6-phosphate. Then that glucose could be stored as glycogen. Let's say you're just resting, you've had a meal, you're not exercising yet. Much of those glucose 6 phosphate molecules can be converted and stored as a large glycogen molecule. So that's glycogen synthesis. And the enzyme glycogen synthase is responsible for creating and storing glycogen. Or if you're exercising, that glucose coming in through the glucose transporter there in red, the enzyme hexokinase will again tack a phosphate onto it at the 6th carbon position, and then through a series of 10 enzymatic reactions, this is called glycolysis, we can produce ATP from the glucose 6 phosphate. Okay, so that glycolysis is going to be occurring during high intensity exercise, breaking down that glucose coming into the cell through 10 enzymatic reactions and creating ATP so that we can keep fueling our muscles to contract. This is the pathway of glycolysis. I am not going to go over all these enzymatic reactions here. It's a pathway involving 10 to 12 steps, various enzymes. But glycolysis is the breakdown of glycogen or glucose in our cells. And it begins out in the cytoplasm of the cell. And it's this coupled and linked series of enzymatic reactions. So you follow all those gray arrows. It's stimulated during intense muscular activity. So rising levels of metabolites that build up in our muscle cell as we're exercising, like ADP, AMP, inorganic phosphate, these all build up as ATP is broken down, as we're releasing energy, as our muscles are working. Also ammonia, and muscles tend to become more acidic because we get a bunch of hydrogen ions released in muscle as they're contracting. So all of that stimulates this pathway, okay? If we have a lot of ATP already available in our muscles, this pathway is inhibited. Also, if we have a lot of stored creatine phosphate, citrate, free fatty acids, all of these are usually present at rest, so this pathway is blunted. We don't need to be breaking down a lot of glycogen and glucose, because if we have a lot of ATP already formed, we don't need to be forming more of it from this pathway. Plus, at rest, we tend to be relying more on fat as a fuel source versus stored muscle glycogen or glucose, right? So basically what happens in this pathway, if you look at the top on the right-hand side, is starting with either muscle glycogen, right, carbohydrate stored in the muscle, or glucose, which is a 6-carbon molecule coming into muscle from the blood. We have some enzymes that start breaking that down, and we convert, you can see where that pathway divides. We eventually get 2 3-carbon sugars formed called glyceraldehyde 3-phosphate, right? So we go from a 6-carbon glucose, or we break down glycogen in the muscle, but eventually it all funnels down and gets converted to glyceraldehyde 3-phosphate. 2 molecules of that. That's why you have 2 sides. And those are 3-carbon sugars. They go through further enzymatic reactions, and then you see we start producing ATP in green. On each side of that pathway, you see 4 molecules of ATP that are produced there. And then at the end of the pathway, we have 2 molecules of pyruvate formed. Now, depending on exercise intensity, if we're exercising at really high intensities, that pyruvate will get reduced to what's called lactate. At lower and more moderate exercise intensities, where this pathway isn't really cranking and the glycolytic flux is less, it's not really working overtime, that pyruvate will build up more slowly, and instead of getting converted to lactate at lower and moderate intensity exercise, it will enter the mitochondria and go through what's called the Krebs cycle. So the pyruvate enters that mitochondria, gets converted to acetyl-CoA. Then there's the electron transport chain involved and a whole bunch of other processes I won't talk about. But ultimately, the Krebs cycle in the mitochondria helps metabolize that pyruvate, and we ultimately produce more ATP energy for our cell through that process. Okay. Now looking at this graph. This is looking at just overall exercise bioenergetic pathways, how we produce energy during exercise. Okay, over time. All right. So the y-axis is the percent of total energy coming from these different energy sources that I'm going to talk about, and then across the x-axis, the horizontal axis, we have exercise of different durations. So exercise lasting 10 seconds, 30 seconds, 60 seconds, 120 seconds, out to beyond 2 minutes, out to several hours. Okay. So if we look at, you see free ATP there in yellow. Okay. That yellow line in our muscle cells, we have adenosine triphosphate. Okay. And we only have enough of that available for about a couple seconds of high intense exercise. When we start exercising very intensely, our ATP levels start to drop because our body starts splitting off a phosphate group from the ATP molecules and releasing energy to fuel muscle contraction during high intensity exercise. We only have enough ATP available in the muscles for a couple seconds, so that has to be replenished. We've got to replenish that ATP with these different energy systems and different energy pathways. So if we sprint then, let's say for 10 seconds, or we do a weightlifting set, that's a high intensity activity. We can replenish our ATP levels by using what's called the phosphagen system. This is where we take stored creatine phosphate, right? This is creatine that's stored and has a phosphate tacked onto it in the muscle, and we release the phosphate from creatine, and that phosphate can be used to convert adenosine diphosphate back into adenosine triphosphate, ATP. So creatine phosphate acts as a phosphate donor to help build more ATP and fuel high intensity exercise like sprinting, weightlifting, anything that's high intense and lasts, say, 10 to 15 seconds. Okay. Now, for exercises that are high intensity and lasting anywhere from 10 seconds, 30 seconds, 60 seconds, 120 seconds, even longer, you see really kind of this peak and then flattening of the peak for power and short duration exercises. This is where, maybe it's a 400-meter sprint. Maybe you're running an 800-meter sprint. This glycolysis pathway in purple is also important for prolonged endurance exercise of, several hours. Okay, so this glycolysis pathway you see in purple can provide energy. It really peaks in under 120 seconds, but it can be active for hours, just as long as you have enough glycogen in the liver to break down, enough glycogen in muscle to break down through glycolysis and make ATP. That glycolysis pathway can be active, right? So again, glycolysis, I just talked about that pathway. It's active in muscle. You break down glycogen or glucose coming in from the blood through those series of enzymatic reactions. You form pyruvate. Also in that process, you're forming ATP to keep fueling muscle contraction, right? It's all about using these different pathways to keep your ATP levels elevated so you can keep exercising. It's just at different exercise intensities and different points in time, one of these pathways may be more active than the other. Like the phosphagen pathway can really only supply ATP for the first 10 seconds. Glycolysis, assuming you have adequate muscle glycogen stored, can supply ATP from the breakdown of glycogen for an extended period of time. Okay. And then oxidative phosphorylation. This is the pathway in blue where from rest and really at lower intensity exercise levels and endurance exercise, this pathway is more active. So low intensity exercise for a prolonged period of time, we use this oxidative phosphorylation pathway. So this is the pathway where we can break down fatty acids through what's called beta oxidation. They're broken down further in the mitochondria and we produce a lot of ATP. But this pathway, breaking down fats for a fuel source, it's more slow, and the rate at which we can make ATP is slower. So therefore we have to exercise at a lower intensity, but we can maintain that intensity for a long time because we produce a lot, many, many more ATP using that pathway than, say, glycolysis or the phosphagen system. So that pathway is important for prolonged endurance activity, along with glycolysis. It's just that glycolysis, we can run short on glycogen after several hours, whereas the oxidative phosphorylation pathway, if we're using fat in that pathway, we can sustain lower intensity exercise for a really long time. That's all I have for this episode. I know I covered a lot. Your brain's probably swimming. But I hope you have an understanding of how carbohydrates are regulated in different bodily states, the fed state, the fasted state, and during exercise. Hormones play a huge role. Intensity of exercise plays a role in duration of exercise as far as what are we going to use, carbs or fat for fuel. And if you have any questions for me about the KU Exercise Science Program, you can call my office phone, 913-897-8516, or you can email me, jtaylor@ku.edu. And as always, thanks for watching this episode of Fitness Fights.